Mendel's laws help us explain how we get new combinations of alleles in zygotes. Punnett squares with monohybrid and dihybrid can help us look at probability, and pedigrees help us look at patterns of inheritance. So when we think about Mendel and his P's, not only is he making observations, but he's doing experiments. And with those experiments, he's collecting data. Now, Mendelian genetics can often be oversimplified way of looking at inheritance because as the traits Mendel looked at, every single one of these traits, there's only one gene that determines what the trait is. And for each trait, there's only two possible versions of it. There's only two alleles. And none of his traits were linked closely on the same chromosome, so they weren't really subject to crossing over or recombination. So as we go here, his laws of independent assortment we've talked about a little bit. So when the homologous chromosomes line up, maternal and paternal is randomly assigned on either side. It's not like all maternal have to be on one side and paternal have to be on the other side. You can have them all mixed up. And the segregation part is that with each of these, again, they get split up so that the maternal and the paternal ultimately end up in different and isolated gametes. So again, with Mendel, he has these initial pea plants that he's really controlling their breeding and their production. So he has what we call the P generation or the purebred generation. So over and over and over and over, tall only gets tall, tall only gets tall, tall only gets tall, and short only gets short, only gets short, only gets short, only gets short. So then in his first cross, what he did is he took all of these purebred tall ones and crossed them with the purebred shorts. And what he ended up with is all one type, all tall. So the trait that shows up in that F1 generation is going to be the dominant trait. But then he took this F1 generation and crossed them, and he ends up with the F2 generation. And all of a sudden, this is where he understands that there are discrete factors that are causing inheritance, because what was original in this P generation can show back up in the F2 generation, even though it was not in the F1 generation. And what he did as he worked through all of this is he raised a tremendous amount of pea crops, and he collected all of this data, and he found certain ratios or probability. And with all of these, in the F2 generation, he saw that almost all of the ratios come out as a 3-to-1 phenotypic ratio, where 3 demonstrate the dominant trait and only 1 is the recessive trait. So sometimes, though, if you do not know, again, we have P crosses and F1 crosses, but you can also have test crosses. If you would just have a tall pea plant and not know its genealogy, how would you know its genotype, whether it would be heterozygous or homozygous dominant? So you can cross that with a genotype that you know. So if you're using these dwarf varieties, you know that the genotype is little t, little t. So if you cross them and you end up getting a 50-50 cross here in the next generation, where half of them are tall and half are short, you know that original one had to be big T, little t, because the only way in the offspring you could get small is if this plant was a carrier. So again, similar situation, you could do a test cross, and if you ended up with all tall, you knew that tall plant had to have the homozygous dominant genotype. So as we start thinking about this and what's going on, we're going to transition into pedigrees a little bit here. So when you look at pedigrees, you have generation numbers, you have marriage lines, the offspring that are produced, and then as these lines come down, these are all the siblings. We also have the male is the square, the female is the circle, and if they have the trait, they're going to be shaded in. So when we start looking at these autosomal pedigrees, an autosome is within humans are the 22 other chromosomes that determine traits besides the sex chromosomes. So whatever the trait we're looking at, it is not found on the X or the Y. So our next thing after we look at this and we understand a little bit about pedigrees is to determine if the pedigree is representing a dominant or recessive trait. And there's some two patterns that will tip us off whether the pedigree is dominant or recessive. So in recessive pedigrees, the affected individuals will be recessive, which means that anything that's shaded in has to have the little a genotype. They're unaffected. We know they have to have at least one big A. And by knowing what this is, we can a lot of times deduce what the other non-shaded ones are. So the big key here is that if the child has it in recessive pedigrees, but parents do not, you know it's a recessive pedigree. So if you try to imagine what that would look like in a pedigree format, it would look like this. So the child has it, but the parents do not. So again, the child would have to be little a, and both parents would be heterozygous or carriers. So again, we have these two main patterns, and this one is not for an autosomal recessive pedigree, because if both of these had it and they were recessive, their child would have to have it. So this pattern will never show up in autosomal recessive pedigrees. But if you see this pattern, you will automatically know that it's autosomal recessive. So again, when we look at this, two affected parents will always have affected children, and that's why this does not work. We'll get into some sex-linked pedigrees, but a lot of times with these autosomal recessive, a lot of times males and females will be affected with equal frequency. When we look at dominant pedigrees, in a dominant pedigree, the shaded ones are going to have the dominant trait. Again, they could be heterozygous or they could be homozygous, but now we know that all the unaffected ones, if they're not going to have it, they have to be homozygous recessive genotypes. So the pattern with this one, the parents have it, but the child does not. So if we imagine this, it would look like that. The parents have it, but the child does not. And again, this we know is recessive, homozygous recessive, so the parents would then have to both be heterozygous, and they could create an individual that does not have it. So again, when we're looking at the two main pattern types that helped us tip off with the pedigrees, again, this one would not work because with a dominant pedigree, this shaded one would have a big A. If both parents are little a, there's no way a child could get a big A. But this is the pattern that we're looking for to tip us off. So when we're looking at this, again, affected children will usually have an affected parent. Two affected parents can produce an unaffected child. And again, that is going to be the thing that tips us off for this. And again, two unaffected parents will not have affected children. This will not work with autosomal dominant pedigrees. So when we're thinking dihybrid, we're going to get into some dihybrid Punnett squares now, transitioning from our pedigrees. And dihybrid just means that there's going to be two traits. And those two traits we're going to assume are on separate chromosomes. So as we go back through here, again, we have homologous diploid chromosomes. So they have backup copies, right? Before we can start meiosis, we do need replication to take place, and we have to create sister chromatids. Now, this idea of how many chromosomes are there, when we create sister chromatids, we have twice as much genetic material, but we still have 2 pairs of chromosomes. And that's something to keep in mind. So when we review meiosis 1 and meiosis 2, meiosis 1, we mix up and divide the homologous chromosomes. We have tetrad formation. Everything's lining up here in metaphase, but in prophase, we might have some crossing over, but that's for another topic. Anaphase, they get pulled apart. And at this point, through meiosis 1, these are now haploid. So we go from diploid to haploid in meiosis 1. But in meiosis 2, we divide the sister chromatids, just like we would in mitosis. But then ultimately, we go from diploid to haploid. We have this meiosis 1 getting us to haploid, and meiosis 2 dividing the sister chromatids. But again, here, we have 2 versions of chromosome 1 and 2 versions of chromosome 2 here. But then at the very end, we only have 1 chromosome 1 and 1 chromosome 2. So we go from diploid to haploid. We can also look at this from a Mendel perspective when we have our P generation. Now we're assigning letters to traits and the genes on the traits. So again, this would be a homozygous dominant P generation. It would create these gametes through meiosis 1 and meiosis 2. All of the gametes are going to have the dominant characteristics in them. The other P generation here is the homozygous recessive. And again, through meiosis 1, meiosis 2, the gametes are going to be haploid, and they're all going to have the recessive alleles. Now, as you cross these, you're going to have 1 gamete from each, and ultimately, through fertilization, you're going to get back to diploid for that F1. When we look at the F1, these gametes can all be variable. So we can mix and match which L goes with which B. So we get these, again, haploid gametes, but they're all a little bit different. And when we have 2 F1s, again, fertilization is random. However, we end up with this phenotypic ratio of 9 to 3 to 3 to 1 in the F2 generation. So if we look at that for probability perspective, if we had this genotype for the parents, again, when we form our gametes, we could, through meiosis, put this B with this L. Or we could put this B with that L. We could put this B with that L or this B with that L. So we have all these different possible combinations of gametes because of Mendel's laws of segregation and independent assortment. So again, on the other side of that F1 generation and these F1 gametes, now as we put together the zygotes, we look at the probability of all of the new allele combinations. And from a phenotype perspective, we know in this F1 generation, we're going to get 9 to 3 to 3 to 1 when it comes to phenotypes. So again, when we start thinking about this with dihybrid, we have 2 traits, the color of the pea pods and the height of the plant in this example. They're going to create gametes. Those gametes are going to get crossed in the P cross. We're going to end up with a heterozygous F1 generation. If we do the F1 cross, we're going to end up with this 9 to 3 to 3 to 1 phenotypic ratio in the F2 generation. So again, we can also have test crosses with dihybrid. So now we're looking at fruit flies with long wings and gray bodies, and those are the dominant alleles. We could cross it if we don't know what genotype is for this dominant allele fruit fly. We can cross it with a recessive allele fruit fly, then we would know exactly what the genotype is for this, because the only way they are demonstrated is through homozygous recessive. So again, we know that this has to be the genotype for this phenotype. To try and figure this out, if we cross these two and they end up like this, we know that the genotype for this individual is going to be heterozygous. Again, if we crossed it but got all of the offspring that look like the dominant phenotype, we know that this individual's genotype has to be that. So again, Mendel's law help us understand about creating new combinations of alleles in the zygotes. We can look at Punnett squares, both monohybrid and dihybrid, to understand probability, and we can look at patterns of inheritance with pedigrees.